I'm Becky Howard from PGRO, and I'm going to talk to you now about some of the research that's been done on post-crop nutrition, and also give you a little update at the end of the presentation on a couple of our projects around sustainable pulse production. So we start by thinking about the importance of nutrients in pulse production. We know generally that more P and K has been removed from arable rotations than is returned. And over the last 40 years, I suppose, since the early 1980s, we've seen significant declines in P and K applications between 50 and 65% in overall in arable rotations on tilled land. So across the board, indices are now below target on 25% of UK soils tested in the UK. And we know, of course, that P and K levels in pulses impact quite strongly on nitrogen fixation, which is really important because that is how pulses provide their own nitrogen. On the other hand, though, when we look at sulfur, which is another key nutrient for pulses, we can see that applications have increased significantly since 2004 by around 90% actually, and there's one significant reason for that, and we'll talk a little bit more about that in a few minutes. So when we look at what the nutrients do for pulses, it's quite important. P and K in particular are implicated in nitrogen fixation and root and nodule development, so very important in pulses, of course. As I said, that's what's providing the nitrogen for the plant itself. Potash, production of sugars and proteins, water transfer in cells, photosynthesis and nutrient use, particularly N. And then phosphate, we're looking at energy transfer in the plant, the production of sugars and proteins, as I said, root and nodule development, photosynthesis and respiration. But phosphate does need efficient mycorrhizal networks in order to be taken up effectively. And phosphate in particular does give some disease tolerance particularly in beans, actually. So when we're looking at chocolate spot, phosphate deficiencies can lead to significantly higher levels of chocolate spot, but also standing ability, so the turgidity of the cells in the plant. Manganese I'll talk about because it has specific implications, particularly in peas. Generally, it's implicated in photosynthesis, biotic and abiotic stress tolerance, and nitrogen metabolism. And sulfur important for protein and amino acid production, chlorophyll formation, and again, nitrogen fixation. So, and sulfur is easily leached. But of course, when we think about nutrition, and there are other nutrients, of course, that are important, but these are the ones I'll focus on today a bit. When we look at uptake, we know that it's very dependent on soil health, particularly organic matter content, biological activity, soil structure and aeration, and specifically pH as well. We have done quite a lot of work over the years. The first project I'm going to talk about really is looking at phosphate in peas in particular. So a few years ago, we did work looking at critical P levels in vining peas. Some of this can be perhaps transferred into combining peas, and this was done across multiple years at multiple sites each year to give us a broad view of how and what levels of P are required in order for peas to function at their best. We looked at two different types of P applications. So firstly, we were looking at stabilised P applications. So this was applied as TSP before the previous crop. And we were looking at sites that had a low inherent P content, so at indices of 0 and 1. And then we were looking to raise those indices to 2 and 3. And we also looked at fresh P applications. So these were applied immediately before drilling of the vining peas themselves. And then we compared across the plots. One of the things that we saw pretty quickly, I suppose, is the impact that phosphate has on establishment and vigour at early growth stages. And you can see from these images here where we have a low index at 1 for P We get poor establishment, low vigour, and then when we look at an index P, which is high, of course, and it's slightly above what we would normally recommend generally in an arable rotation, but we can see the impact this has in peas quite significantly in terms of vigour and establishment. So what we found from the project overall is that we are highly likely to see yield losses at soil P indices of 0 or 1, and that was up to about 7% in vining peas at an index 1 compared to 2. So the general recommendation from that project was to maintain soil Olsen P at index 2 for optimum yield in most situations. In vining peas, particularly in high yield potential situations, we did find that an index 3 could be more appropriate, giving us an additional benefit. And of course, in a high value crop like vining peas, a 4% yield benefit transfers into a reasonably significant economic benefit. We do, however, need to think about the risk of soil P losses to water. So this is through erosion of soils into water that leads to eutrophication. And that can be quite a significant risk because what might seem like a small amount of P losses of 2 kilograms per hectare can actually lead to quite a significant eutrophication risk. So that does need to be kept in mind. And actually, if we're looking at longer term raising of Olsen P indices, certainly in terms of economic returns, that can take between 1 and 4 cropping years, depending on the yield potential of that crop. But we did find that relatively small doses of fresh P as TSP can give a yield benefit in some cases, and that can be at a reasonably low cost. There's been all other work done on the effects of phosphate on nodule formation in beans in this case. So we can see here, and this work was published by the PDA, the Potash Development Association. And when we look at reasonably small doses of phosphate here, so 20, 40 and 60 kilograms per hectare, we can see that there is a benefit in terms of nodulation. So number of nodules per plant in the blue columns. And then as we move across to where we've applied phosphate, we can see the percentage increase in the orange bars, the increase in nodule weight in the green bar, and the percentage increase in nodule weight as well. So gradual increase in terms of nodule production when phosphate's applied. And I suppose the question we ask ourselves really is, how low can you go? So when we look at starter fertilizers that include P as part of that starter fertilizer, we have tested a couple of products in the past. One is primary P, and you can see here it does have a small amount of nitrogen in it. So Nmax in peas and beans is 0. So technically, although it's only 1 kilogram, when you look at the actual rates of application of nitrogen, we should not be doing that. However, we looked also at a product called Microstar, which doesn't contain nitrogen. And they both contain 4 to 4.5 kilos of phosphate in the starter fertilizer. And we looked at those across multiple sites in multiple years at a 3/4 rate, a whole rate, and then 125% of the recommended rate. And that is 7.5 kilos, 10 and 12.5 kilos of each product. When we look at the results of that, we can see there are some general trends, I suppose, that we would say came out of those trials. And these results are looking at all trials across all years. Although they are not statistically significant, you can see a slight trend certainly for a yield increase when we apply the slightly higher doses of the primary P and the Microstar. What's interesting, I suppose, is, although again I say it's not statistically significant, when you look at the higher dose of the primary P, and that's the one that contains N, that small dose, you can see nodulation is slightly lower than when we put in a 10 kilograms per hectare rate of primary P. And we do know that low levels of nitrogen in soils can sometimes suppress nodulation in peas and beans. But actually, when we looked across all the years and at all the sites, we can see that there were other effects that were stronger actually in terms of nodulation and yield. The time of drilling was a strong factor, particularly on yield. And we can see for nodulation, the early and mid timings, nodulation was better. And an early timing was the end of March, the mid timing was mid-April, and the late timing was early May. So you can see those early May drillings are not forming as many nodules per plant. The effect on yield equally significant. So we can see that mid timing for vining peas in April is having a better effect on yield compared to the early and late timings. But the year effect on yield is also important. And I think it's important to remember this as we carry on through this presentation, really, because I'll be talking in a little while also about biostimulants that contain trace elements and nutrients as well. But year effect is quite important in terms of nutrient uptake and the effects of weather and conditions on nutrient availability and uptake by the plant. Potash is a deficiency that I would say is reasonably easy to recognise. So when we're looking at phosphate, which we've just been talking about, we can see that that does have these impacts on early establishment and vigour. But potash deficiencies in beans can be seen, and you can see them in the images here as this purpling around the edge of the leaves, which over time becomes brown and then necrotic. So you get this blackened leaf edge. The other thing that's really clear when you have a potash deficiency in beans is the shortening of the internode. So the nodes are very close together and therefore the plants are extremely stunted. And this is one of the most important of the major nutrients for pulses. They have a high requirement for potash in order to form root nodules and root systems, and any deficiency is likely to cause yield depression if it's not corrected. And most of the time when we're talking about P&K, we're talking about pre-drilling applications. Potash has a very sort of close relationship with magnesium in soils, and therefore the impact of high magnesium levels can also reduce potash availability. And that in turn then leads to reduced nitrogen utilisation. So it is important that we think about this and that we get this right. And it's one of the deficiencies that we do see coming through our plant clinic here at PGRO more frequently than we would like to. So work has been done looking at the influence of potash on activity of rhizobia. So we're looking here at soil potash, and we've got a comparison here done between a low K soil, medium K soil and high K soil, and that's an index of 1, 2 and 3. So we can see we're using our low K soil as our control, if you like, where we've got 100%. We're using that as 100% of nodulation and nitrogen production, and then comparing the medium and high levels to that. So we can see we do get increases in nodulation, so both in size of nodules and number of nodules and production of nitrogen in the plant, the higher the level of potash in the soil. But when we look at bean yield responses here, so this is in field beans, and here we're looking at two different soil types or soil levels of K. The first is at index 1 on the left, so that's a low K soil, the two bars on the left, and then we're looking at an index of 2- on the right. And then there's a 0 application of potash compared to 53 kilograms per hectare of potash applied. And what you can see is that it is responding, the beans are responding quite well in terms of yield on that low potash soil where potash is applied. But when we get to what you consider to be a standard sort of adequately a soil with adequate potash, the bean yield is not responding when you add an application of potash. And we have also found this in data that we've generated at PGRO. So we had a look at potash amongst other products that are included in this trial at 40 kilograms per hectare applied as muriate of potash, and also at 80 kilograms per hectare. compared to the untreated plot, which you can see as the red bar there. And you can see there are no significant differences in yield between any of the treatments, and we've got a yield variation between about 6.2 and 6.6 kilos per hectare. So we can see here, and at this site, I should add that the potash index was at an index 2, so it's adequately supplied, and therefore we're not getting any yield response in that situation. But when we look at peas, both combining peas and vining peas, which were tested at the same time on the same site, so adequately supplied with potash at an index 2, we can see they are responding quite strongly to potash applications at reasonably low doses actually. So it's not expensive. At current price, we're looking at about 12 to 25 pounds per hectare to apply 40 or 80 kilograms of muriate of potash. And in combining peas, that is giving us an advantage of between 35 and 75 pounds a hectare, which is reasonably significant. And when we look at vining peas, the higher value crop, we can see even though we're looking at a reasonably low yield impact here, it has a significant effect on the economic return. Sulfur we talked about briefly. You can see here, and this information's from various sources, but we're looking at emissions really over the years. So we can see through the sort of industrial revolution era that we had high levels of sulfur deposition, and this has significantly declined. And currently we're in a situation where there's almost negligible sulfur deposition in the UK. And this, of course, is a good thing, but it does impact on how much we need to apply in terms of crop requirement. So the work that we've done over the years, looking at using polysulfate actually at 3 different rates in both vining peas, combining peas and spring beans, we can see that the influence of polysulfate is quite strong again in vining peas. So they're responding to the sulfur up to about 150 kilos per hectare of polysulfate. We can see that the combining peas from the left figure, and that's the orange bars, do respond, but they don't. There's no differences in their response to the different levels of polysulfate. So you're getting a good response at 100 kilos per hectare of polysulfate. But again, beans not as responsive here, so we're not really getting any major differences between the applications and the untreated in this trial. But we looked at nodulation as well, and we can see that reasonably small applications of sulfate are actually giving a benefit in terms of nodules per plant and nodule weight from that figure on the right. I suppose the other key nutrient that I want to talk about is manganese, and it has a very specific, there's a more specific reason why we talk about this, particularly in peas. So manganese deficiency is reasonably easy to identify in the plant. It leads to yellowing, interveinal yellowing, and also there's an easy way to remember to distinguish manganese deficiency from magnesium deficiency in this case. So the symbol for manganese is Mn, and we use the sort of short, so the margins are not green in this case. So margins are yellow, whereas in magnesium deficiency, the margins are green. So you can see from that plant there that we've got yellow leaf, and also actually you tend to often see it in stripes in a field, related to compaction in the field or the tractor wheelings as well. So what we generally find is that manganese deficiency is common on soils of pH 7 or above, so on alkaline soils, or those that have had heavy lime applications, also quite common on organic soils, and particularly in wet soil conditions. And often we would say when it comes to a recommendation for the solution for this problem is you we normally recommend 2 applications at least in peas where a manganese deficiency is known. In wet conditions, that can often be 3. Now, the reason it's so important is because we see this disorder in peas called marsh spot. So in the bottom right there, you can see an image that's showing necrotic spotting in the centre of the pea. And you get this very brown granular mark in the centre of the pea. So this affects the quality for processing. Most of our peas are going into high value human consumption markets, and this is unacceptable. So it's very important that we treat it. It also affects quality for seed production as well. So when we have marsh spot in seed, we can see that we get these abnormalities in the pea that's growing from that seed, and it causes really a sort of barren seedling, production of multiple shoots to try and compensate for that. And those seedlings are abnormal and won't produce a normal pea plant. It's reasonably easy to rectify in the field, but I think the important thing to emphasise is that we should be using formulations that contain 32% manganese sulphate. If we look at other formulations, and there are multiple types of manganese that can be applied onto crops, we can see that they're not always as effective. So if they're a lower percentage of manganese sulphate or manganese, then you need to apply more, because what we're targeting, and you can see on that bottom row of that table on this slide, we're targeting 5 kilograms per hectare at each application. So at 32% formulation, you'll be looking at a lower dose, basically, in order to achieve that. And what we'd say is you will need to apply if foliar symptoms are seen. So if you're seeing symptoms of manganese deficiency in the crop, then you should start applying manganese immediately and repeat at 10 to 14 days. If you don't see symptoms, but you know that you historically suffer with manganese deficiency or marsh spot, then you should be treating at first pod in order to prevent marsh spot. And as I said, you need to think about, in wet seasons, applying a 3rd application. So moving on to biostimulant products, and of course, it's difficult often to define what a biostimulant is, but overall, they contain substances that stimulate the natural plant processes. Now, that can be for multiple objectives. One can be increasing tolerance to environmental stress or disease, for instance. It could be to improve soil conditions and availability of inputs, or it might be to improve the quality and yield of the final product. So I think really what I'd say before we talk about some of these products is it's probably quite important that we determine what our objective is before we think about what product that we're going to use, and actually really think hard about how these fit into an integrated crop management system, because often with biostimulant products, whichever one you're using, they won't be a total solution. So you're almost stacking your solutions in order to achieve whatever objective it is that you're looking for. And there are multiple types of biostimulant: fulvic acids, trace elements and micronutrients, peptides, seaweed products, and biological products, so fungal and bacterial products as well. So and as well as rhizobial products to stimulate root nodule development. We've tested most of these types of biostimulant in trials, and I'm just going to talk about a few of them today. We did trials over multiple years at multiple sites and in all of the crops that we work with, so combining peas, vining peas, spring beans and winter beans. I'm only going to talk about yield now. We do look at things like crop establishment, disease development, and other indicators of performance of the crop, but we'll just talk about yield in terms of these trials over a number of years and in all of the crops. And some of them are starred, and I'll tell you why they're starred in a few seconds. So I'm comparing them to the untreated here, which is the red line across the middle of this chart here. And we can see that some of them are giving reasonably good yield benefits, up to about 5% yield benefit, and these were statistically significant. So we can have reasonable confidence in these results over a number of years. And included in this trial are nutrient and trace elements, seed treatments, soil treatments, and foliar treatments. We've got Serenade in here, which is bacillus, which is a bacterial treatment, pre-stop at the end, treatment 13, which is a fungal treatment, which is glycladium. And the ones with the stars on them are the ones that contain phosphites. So phosphites are a product that are applied either to the seed in startup, takeoff, multimax and kickoff are all seed treatments. Force is a foliar treatment, and this case was applied twice before flowering. And I think it's important to just talk about the role of phosphites. They are not providing phosphate to the plants, but they enable the plant to take up phosphate perhaps slightly better, and can give some resilience to disease in crops as well. And overall, I would say they do seem to lead to slightly better performance of crop performance. But we do need to be careful because they're not broken down by the plant. So they leave residues of phosphonic acid and phosphatile aluminium, which are picked up in crop products. So if we're using them, it's really important to use them earlier as a seed treatment or before flowering as a foliar treatment. But we can see that we are getting some benefits from some of these products. We work on biostimulants every year, actually. We've done so for many years. And overall, I'd say a summary of the biostimulant work is that it is a bit of an umbrella term. We don't think it's specific enough. And it's important really to consider why you're using it or why you're choosing a particular biostimulant product. What is your objective to use it? And then we can categorize them really into seed treatments, soil treatments, or foliar treatments, or we can categorize them by the target that we're looking at. So is it establishment, disease resilience, final yield? So we know that the biopesticide applications and the phosphite-containing biostimulants gave us increases in yield and improved resilience to disease, as did some of the macro and micronutrient products. But overall, they are very inconsistent, and we continue to test new products, but it's important to think about the benefits of using any of those products over a number of years, because they react very differently to the different conditions that we get in each year, and the cost benefit might not always be good in any given year. So do include them if you need to as part of an integrated crop management program, but do not expect them to give you an entire result in terms of giving you a benefit to yield or improved resilience to disease. So finally, I'm just going to give a quick update on the two projects that are running as part of the Farm Innovation Programme funded by DEFRA. The NCS project has now been running for almost 2 years, and we're coming up to our second anniversary. So we've had a whole year of both the crop trials that are included in this project run by our Pulse Pioneers, and also the animal feed trials that are included in the project. And there's a number of really important findings that are coming out of this project now. So one is that the benefit of pulses in rotations, certainly regarding the provision of nitrogen within a rotation, are probably more than we originally thought, but they are quite variable. The original aim of this project, of course, was to increase the proportion of pulse crops in the rotation to 20%. The key objective, of course, is to reduce climate impact in arable rotations. And a lot of work has been done by the Farm Carbon Toolkit and the animal feed partners, SRUC, MacArthur Agriculture, to look at whether we think we can still achieve that level of carbon emissions reduction by increasing pulses. And there's very clear evidence that that is possible. And in fact, that we'd significantly underestimated how much benefit can be obtained by increasing pulses in rotations. And that, of course, is gained through the reduction of fuel costs in pulses, the direct reduction of nitrogen applications in pulses, as they don't require nitrogen applications. The indirect reduction of nitrogen in rotations as well, certainly to the following crop, and also actually the reduction of soya imports into the UK if we can replace soybean meal in animal feeds, which a lot of evidence from the animal trials is showing that is certainly possible. Moving on quickly to show that our original estimation of the amount of reduction of carbon emissions was 1.5 million tonnes per annum. So that was estimated at about 50% of the maximum potential reduction of CO2 for UK agriculture. The work that's been done by Farm Carbon Toolkit is showing that actually, if we would get to 20% of arable rotations in pulses, that could be up to 3.4 million tonnes per annum, which is huge. Finally, I'm just going to briefly talk about a new project called Answers. This project is specifically looking at nutrition in field beans, and this started a few months ago. So we're in our very first year of trials. We're working with MPZ as the lead partner, Yara, and the University of Lincoln. And again, this is in the Farm Innovation Programme funded by DEFRA and supported by Innovate UK. The purpose of this really is to try and fill some of the gaps in knowledge about nutrition in field beans, and there are a few gaps. So this includes applications of P and K as soil applications, the use of seed treatments and the micronutrient seed treatments, as well as magnesium in soil applications, and then foliar micronutrient and trace element applications. An important aspect of this is also to look at how our winter and spring bean varieties interact with those nutrients. So this work has just started, and we hope that we'll have outcomes from the first year of trials by October 2025, and those will be published. So that's me done, and thank you very much for listening. And you should be able to find summaries of both NCS and the Answers project on the Pulse PEP website, which is part of the Farm PEP website, which is provided by ADAS. Thank you.